Hi, everyone. My name is Julia Hajar. I'm a third-year PhD candidate in the Population Health Program here at the University of Ottawa, and I study the impact of endocrine disrupting chemicals on maternal fetal health. So I'm here today to talk to you a little bit about nuclear receptors and endocrine disruption, and we're going to cover a few topics. So I hope you find this presentation interesting, and please let us know if you have any questions. So just some learning objectives before we start. So we'll talk a little bit about nuclear receptors and their basic mechanism of action, major ligands that are involved in nuclear receptor signaling, some definitions, so the definition of endocrine disrupting chemicals, and an overview of how they act to disrupt hormone signaling. We'll also talk a little bit about how endocrine disruptors impact the HPA axis or main stress pathway, some common environmental exposures to EDCs, and some evidence for how they disrupt our health. So just an overview of nuclear receptors. So these are transcription factors that are essential in many activities in the body, including embryonic development, maintenance of differentiated cellular phenotypes, metabolism, and cell death. So unlike other types of receptors, which may be found on the cell surface, these proteins are intracellular transcription factors that can directly regulate how genes are expressed in response to lipophilic molecules, such as hormones. So steroid ligands, so some of our hormones can bind to these receptors. So for example, the estrogenic hormones will bind to estrogen receptor alpha or beta. Progesterone will bind to the progesterone receptor. Androgens will bind to the androgen receptor, thyroid hormone to the thyroid receptors, and glucocorticoids to the glucocorticoid receptor. And we'll focus a little bit more on this particular receptor in this lecture. And then non-steroidal lipophilic hormone ligands can bind to receptors as well. So these could be things like vitamin D, vitamin A, and fatty acids. And there's also orphan receptors, which have been found, but no ligands have been identified yet. So here's just a general mechanism of action of nuclear receptors and how they work. So nuclear receptors can be found in the plasma membrane or in the cytoplasm. So hormones can cross the plasma membrane and bind to cytoplasmic receptors. When they're bound to a ligand, they then change conformation and lose affinity to the inhibitor. Then they can migrate to the nucleus and bind to DNA, where they can turn transcription of genes on or off. And some endocrine disrupting chemicals can bind directly to these receptors. So they can act as either agonists or antagonists, and they can enhance or inhibit the effect of a hormone. So ultimately, the cellular response will be to change gene transcription. So let's talk a little bit about estrogen receptors. So estrogen receptor alpha and estrogen receptor beta. So they can overlap in some biological function, but this largely depends on the type of tissue and cell. So for the estrogen receptor alpha, these are found mainly in the mammary gland, the uterus, the ovary, bone, male reproductive organs, the prostate, liver, and adipose tissue. So their more prominent role is in the mammary gland, in the uterus, maintaining skeletal homeostasis, and in metabolism. And for estrogen receptor beta, they're found mainly in the prostate, the bladder, the ovary, the colon, in adipose tissue, and in the immune system. So estrogen receptors play a really important role throughout the body in many different pathways. So endocrine disruptors such as bisphenol A or BPA can bind to both of these estrogen receptor subtypes. However, it binds at a much lower affinity compared to estradiol. So BPA can actually mimic estradiol, which we'll talk about a lot in this lecture, and it can bind to ER alpha to activate rapid extranuclear pathways, drive calcium release, which can then cause changes to cell motility, cell signaling processes, and exocytosis. So it can totally change the activity of the cell. BPA can also act as an antagonist of estradiol by acting on ER beta to prevent downstream signaling to targets such as P38-MAPK, the mitogen activated protein kinase, which is important for signal transduction. So endocrine disruptors can really impact our cells. And I think by the end of the lecture, you'll be able to tie all of these different concepts together to see how nuclear receptors work and how these endocrine disruptors impact their function. So let's talk a little bit about the glucocorticoid receptor, so how it works. So the glucocorticoid receptor is a member of the nuclear receptor superfamily of ligand-dependent transcription factors. So how does it work exactly? So glucocorticoid receptors bound to a ligand initiates or represses gene transcription, like we just talked about. And this can exert effects on thousands of genes. So glucocorticoid receptors can really impact the whole body. It binds directly to DNA response elements and or can physically associate with other transcription factors. So it can promote gene transcription and can drive the initiation, creation of new protein. So glucocorticoids and glucocorticoid receptor and mineralocorticoid receptor binding. So glucocorticoids can bind to intracellular receptors, so the MRs or mineralocorticoid receptors with a high affinity, and glucocorticoid receptors with a lower affinity. So they're lipophilic and they bind to receptors in the cytoplasm, these nuclear receptors. So once they're bound, the activated receptor complex undergoes nuclear translocation, the same as the pathway we talked about a few slides back. Once it reaches the nucleus, the receptor complex acts as a transcription factor and can bind to specific glucocorticoid response binding elements on the DNA. And then transcriptional response to stress and local epigenetic changes can then occur in a cell type specific manner. So I want to talk a little bit about the HPA axis and how this negative feedback loop works. This is important just so we understand how receptors are linked, how they initiate the next stage of the pathway, so how they initiate the release of hormones and how hormones can feed back in this loop and make sure that the pathway is turned off as appropriate to maintain homeostasis. So when the body is in fight or flight mode, there's a stressor present, the HPA axis is activated. So it has three main components: the hypothalamus and the anterior pituitary in the brain, and the adrenal glands, which secrete glucocorticoids. So once a stressor initiates this pathway activation, the hypothalamus will secrete corticotropin releasing hormone, which will travel to the anterior pituitary and bind to receptors there. And once it binds to its receptors on the anterior pituitary, this will stimulate the secretion of adrenocorticotropic hormone, which will then travel to the adrenal gland and bind to its receptors there. And once it binds to its receptors on the adrenal gland, this initiates the release of cortisol, and cortisol will then travel throughout the body and exert its effects. The important part here is that this pathway is turned off as soon as we don't need this activation and the secretion of cortisol. So it's important that the pathway is really conservative and is turned off as soon as we don't need any more circulating cortisol. So cortisol will then feed back into its receptors at the level of the anterior pituitary and again at the hypothalamus. And this will tell the hypothalamus to stop secreting corticotropin releasing hormone and will shut down this pathway so it's not overactive. And we'll talk about this in a little bit, but we'll see how endocrine disruptors can impact the receptors, the glucocorticoid receptors, which are involved in this HPA axis. And we'll see how this can then modulate the way that the HPA axis is regulated. So let's talk a little bit about how estrogenic compounds can modulate the glucocorticoid receptor. So this can happen three ways. So either there can be proteasomal degradation by estrogen ubiquitin ligase. So the GR can actually be degraded. The GR can be inhibited to phosphorylate. So it needs to phosphorylate to be able to be active. So this phosphorylation is inhibited by estrogenic enzymes. And the third way is estrogenic repression of GR expression. So this can lead to a decreased gene expression of glucocorticoid receptor and an overall decreased number of glucocorticoid receptors that are available for binding. So this can also contribute to an upregulated HPA axis and an overactive stress pathway. So let's talk a little bit about endocrine disrupting chemicals again. So these are ubiquitous exogenous substances that can interfere with the endocrine system, as we talked about earlier. So hydrophobic molecules from the environment mimic our body's natural hormones, and our body can't tell the difference, and they disrupt hormonal balance in the body. They can impact reproductive processes, which rely on hormonal signaling mediated by nuclear receptors. Some common examples of endocrine disruptors that you may have heard about include phthalates, BPA, diethylstilbestrol, which is a very potent xenoestrogen, parabens, pesticides like DDT, PFAS chemicals, which are typically found in nonstick cookware, just to name a few. So the impact of endocrine disruptors on healthy growth and development. So we know that an insult during a sensitive period during development may lead to altered programming or tissue structure and function. This can be permanent if these exposures are during critical windows of development, such as during pregnancy or during early infancy. So exposure during these critical windows can result in lifelong health consequences because important organ systems are still developing, the brain is still developing, and they're very susceptible to disturbance by these endocrine disruptors. So this can contribute to the development of later behavioral problems, learning difficulties, altered or delayed cognitive development, cognitive decline, psychopathology, cancer, cardiometabolic or neuroendocrine issues, obesity, and reproductive health issues, just to name a few. So some examples of endocrine disruptors, as I mentioned before, include diethylstilbestrol. So this was originally prescribed as a pill to combat morning sickness and to mitigate pregnancy complications between 1950 and 1970, and it proved to be extremely carcinogenic. So it's DES daughters, so the daughters of women that were prescribed this and who were exposed in utero were at much higher risk of developing clear cell adenocarcinomas, breast, pancreatic, cervical, and vaginal cancers as a result of exposure to diethylstilbestrol during gestation. So another example of endocrine disruptors are phthalates. So these make plastic flexible, and they're also present in personal care products like nail polish, hairspray, perfumes and colognes, shampoos and soaps, and even in blood bags and tubes and medical equipment. Phytoestrogens are a natural source of estrogenic chemicals. These are plant sources of estrogen mimickers. So this could be something like soybeans or chickpeas or tempeh, and even sesame seeds. And pesticides like DDT or glyphosate, which are sprayed on crops, and PFAS chemicals, which, as I mentioned, are typically seen in nonstick pans, and they're also used in firefighting foams. So 10 key characteristics of endocrine disruptors. So they can act as a receptor ligand or as an agonist. They can act as a receptor antagonist. They can modulate receptor expression. They can modulate signal transduction and epigenetic alterations. They can modulate hormone synthesis and transport, as well as hormone distribution and circulating levels of hormones. They can also impact the breakdown of hormones and their clearance from the body. And they can also impact cellular fate. So whether a cell differentiates, proliferates, or undergoes apoptosis. So endocrine disruptors can exert their effects three main ways. The first is that they can competitively bind to receptors. So they'll prevent our body's natural hormones from binding to their receptor. And they can modulate our hormone receptor function. And they can stimulate the production of our hormones, or they can inhibit the production of our hormones. They can change the way that our hormones travel throughout the body, which then affect their function. They can also alter normal levels of inactivity of many hormones, including estrogen or testosterone. So they can also impact glucocorticoid receptor and mineralocorticoid receptor binding. And they can do this a few ways. So they can disrupt the way the ligand binds to the receptor. So the way, for example, cortisol would bind to the glucocorticoid receptor. They can also impact how the ligand receptor complex travels into the nucleus. And they can also modulate the expression of endogenous glucocorticoid receptor or mineralocorticoid receptor genes. So let's get back to this diagram again. So the HPA axis, but this time let's see how these endocrine disrupting chemicals can impact the way that this axis works. So we remember with normal feedback loops, the axis will be turned off when it's no longer needed to be activated. However, when we're exposed to xenoestrogens, so for example, something like BPA, something like heavy metals like lead, this will impact the glucocorticoid receptor levels and amounts. And in turn, this will affect negative regulation. So the feedback loop won't turn off properly. And as a result, we'll have excessive cortisol secreted and circulating throughout the body. So ultimately, this will result in downregulation of glucocorticoid receptors, downregulation of their binding, disruption of negative feedback, and ultimate upregulation of the stress axis pathway. So there's been a lot of evidence emerging in recent years about the adverse effects of endocrine disruptors, especially at low doses. So in 2015, the Endocrine Society analyzed over 1,800 studies on endocrine disruptors and released a report showing how these endocrine disruptors disrupt the function of our hormones. So they provided evidence that these endocrine disruptors are involved in a myriad of health issues, including hormone-related cancers, osteoporosis, diabetes and obesity, immune and thyroid disorders, reproductive disorders, and neurological problems, just to name a few. So we know that endocrine disruptors are quite prevalent in the environment. They're often persistent, so they don't break down. They end up in our lakes and streams and back into our water and into our food, and they're really omnipresent. So it's important to be aware of these endocrine disruptors and how they can impact our health. So there's been a lot of research emerging on the impact of endocrine disruptors on reproductive health and fertility. So we know that endocrine disruptors can modulate important biological processes such as organ development, reproduction, even the immune system, behavior, neurodevelopment. And in recent years, BPA and phthalates have moved to the forefront of research because there's concern regarding their impact during the critical window of pregnancy and fetal development in humans. There's also increased exposure to phthalates in one research study during the early phase of the first trimester of pregnancy has shown to be linked to higher concentrations of estrogen and lower concentrations of testosterone in the fetus. So this can affect normal male development in utero and increases the chances that male babies will be born with abnormal genital development. And female reproductive development, fertility, and the onset of menopause are all impacted due to exposure to these endocrine disrupting chemicals. There's also been a significant decline in male reproductive health in recent years. So sperm count and testosterone levels have been significantly reduced. And semen parameters can be used to measure sperm quality and can help predict male infertility. So poor semen quality has been linked to exposure to certain endocrine disruptors, and this has also been observed in recent years. So emerging studies in humans suggest that there's an association with decreased fertility, lower semen quality, reduced sperm motility, and increased DNA damage to sperm. And in a study by Lee, there was a urinalysis performed of men who worked in BPA-based factories in China. And it was found that those men were 4 times more likely to have lowered sperm counts and more than twice the risk of having lower sperm motility. So detectable levels of BPA in the urine of these men had decreased sperm concentration, total sperm count, sperm vitality was lowered, and sperm motility. A small-scale study out of UCSF reported a reduction in the quality of eggs retrieved for in vitro fertilization. As blood levels of BPA in the women in the study doubled, there was a 50% reduction in the percentage of eggs that were able to fertilize normally. So BPA is at the forefront of research when it comes to fertility and looking at the impact of endocrine disrupting chemicals on fertility. This is just another diagram of some other examples showing different endocrine disruptors that have impacted different parameters, including semen volume, sperm concentrations and counts, embryo quality, time to pregnancy, miscarriage. So because of this increased use of man-made chemicals, with over 1,400 currently deemed to be EDCs, there's been a lot of cause of concern about the impact of these endocrine disruptors that have become much more prevalent in our everyday environment on how they impact fertility. So let's talk a little bit about obesogens. These are endocrine disruptors that can actually influence obesity. So they affect energy metabolism, adipose tissue structure, and lipid and glucose metabolism. They're mainly lipophilic in structure and they bioaccumulate. So it's a destructive cycle because it promotes the retention of other lipophilic pollutant chemicals. So obesogens can act several ways. So they can alter lipid and glucose metabolism, they can alter insulin sensitivity, they can increase the size and the number of adipocytes. They can alter hormonal regulation of adipose tissue and promote adipogenesis. And they can also modulate neuroendocrine control of appetite and satiety. So some examples of obesogens include diethylstilbestrol, which is the endocrine disruptor that I mentioned before. It's a very potent xenoestrogen. Plasticizers, so bisphenol A and phthalates, preservatives like parabens. They're even found in plant sources and phytoestrogens. So things like soy and tempeh and tofu. In flame retardants and in agriculture, so in persistent organochlorines, for example. So we'll focus a little bit on BPA here. So BPA is quite prevalent in our everyday society, everyday life. It's an organic compound used to synthesize epoxy resins. It's found in a wide range of products, including water bottles, thermal paper. So it's even on receipts that you can get after a transaction, plastic toys, can linings, and medical devices. So over 15 billion pounds of BPA is produced per year, so it's quite widely used globally. And the primary exposure routes are through oral ingestion, absorption through the skin, and through inhalation. So it can leach. So the polymer gets broken down through exposure to high heat, exposure to acidity. So if you pour a hot tomato sauce into a plastic container, that's an example of a way that BPA can leach out of the container and right into your food. So you want to avoid exposure to heat and acidity with plastics. So it's also an established reproductive toxicant in rodents. A lot of studies have been conducted looking at low dose BPA exposure in a rodent model. So BPA can mimic estrogen and can thus mediate decrease in the glucocorticoid receptor levels and function and activity, as I mentioned before. So it can reduce GR gene expression, it can lower the number of available glucocorticoid receptors for binding, and can modulate negative feedback loop dysregulation of the HPA axis. So it's really important that we understand how these endocrine disruptors can impact our hormone function, can impact pathways such as the HPA axis, and some reproductive health pathways as well. So it can bind to estrogen receptor alpha and beta. It can also bind to the glucocorticoid receptor, and it's hypothesized to exert glucocorticoid-like action. And in Canada, it was found that the highest concentrations were in children between the ages of 3 to 5 and 6 to 11. And infants had the highest food intake and body weight ratio and the highest BPA exposure levels overall. And BPA is omnipresent, so it was detected in the urine of over 95% of Canadians of all ages, as well as in breast milk, cord blood, and meconium. So it's really present in pregnant women, so it's evident that it's being exposed to the fetus as well, which is modulating development. So this is really an important chemical to study. So the health consequences of BPA include anxiety and depression, ADHD, conduct problems, emotional reactivity, obesity, behavioral regulation, just to name a few. So BPA causes quite a few health issues. So this is exposure during pregnancy, as I mentioned before, is one of the most serious, has one of the most serious consequences because this is a critical window of fetal development, and disruptions during this time period can cause permanent effects because the fetus's brain is still developing. So it's pretty vulnerable to perturbation during this time. And the transfer can occur from the mother to the fetus through the placenta and from the mother to the infant through breast milk. So ironically, it's actually during this most critical time in development that there remains a significant gap in the literature. And in animal research, studies have shown that exposure to endocrine disruptors can cause permanent changes in brain development and behavior if exposure occurs during this plastic period during pregnancy. So I'll talk a little bit about my master's thesis. So the study that I did looked at low dose exposure to bisphenol A. So I looked at gestational and lactational exposure to BPA in rat pups and how this exposure affected their morphometric parameters. So things like their body weight, their body length, and how it affected glucocorticoid receptor gene expression levels in the adrenal gland, which is one of the last component of this HPA axis pathway. So as we talked about with the HPA axis, in rats, there's actually a unique physiological phenomenon that occurs during development called the stress hyperresponsive period. So this occurs to protect the brain during this critical time period so that it doesn't get perturbed by stressors. So the HPA axis is still functioning, but it's really inhibited. So there's low circulating levels of corticosterone and low affinity to adrenocorticotropic hormone. So this pathway is just not really activated during this important period of development. So in my study, I collected samples from rat pups right before this stress hyperresponsive period started and right after the stress hyperresponsive period ended. So I wanted to know if gestational and lactational exposure to BPA perturbed perinatal development in the rat pup. So I carried out a BPA dose series exposure study in timed pregnant long Evans rats. So every day I dosed the pregnant rats with low levels of BPA, lower than what we would typically observe in our environment as humans. And I looked at gross morphology parameters in these BPA exposed rat pups at 2 time points post-birth. So at postnatal day 5, which is right before the stress hyperresponsive period started, and at postnatal day 15, which is right when this stress hyperresponsive period ended. And then I wanted to characterize the molecular effects of BPA on glucocorticoid receptor gene expression levels in the adrenal glands of the male rat pups that I collected. So just a quick recap of what I found. So I had 5 groups of rats. The first was a control group just using a sucrose solution. The second was a group, a positive control using diethylstilbestrol, so that potent xenoestrogen. And then I had 3 groups that had low doses, environmentally relevant doses of BPA. So I'll just talk about the molecular component of the project. So looking at the glucocorticoid receptor gene expression levels. So I had 2 reference genes used in my quantitative polymerase chain reactions. So I had 18S rRNA, which is present in all eukaryotic cells. It's the structural RNA for cytoplasmic ribosomes. So these reference genes are used because we're not expecting anything to happen as a result of exposure to BPA or the dose that you're using. So we want to make sure that our sequences and our qPCR are amplifying regardless of the treatment type. And the other reference gene that I used was GAPDH. So this is an enzyme of the glycolytic pathway. It has high levels that are present in all cells. So I wanted to look at GR gene expression. So what I found in my study was that downregulated fold changes. So this translates to downregulation of glucocorticoid receptor gene expression. So exposure to diethylstilbestrol in my rats corresponded to a lower level of glucocorticoid receptor gene expression. So this was consistently observed in this diethylstilbestrol positive control groups, which is that very potent xenoestrogen, at both age groups, so both at postnatal day 5 and at postnatal day 15. And from what we learned about BPA and endocrine disruptors and how they can impact the HPA axis, this was kind of an expected outcome. And upregulated fold changes were observed in the postnatal day 5 BPA 50 pups, which is just one of my dosing groups, and the postnatal day 15 BPA 500 pups. So we're seeing different effects at different age groups and in different dosing groups as well. So in my reference gene, my 18S rRNA gene, I actually was surprised by this result because it showed that exposure to BPA and to diethylstilbestrol actually impacted the levels in this graph, which I wasn't really expecting. So downregulated fold changes, so a change to gene expression was observed in postnatal day 5 animals in the potent xenoestrogen group, so this diethylstilbestrol group, and then 2 of my dosing groups, the BPA5 group and the BPA500 group. And an upregulated fold change was observed in the postnatal day 5, so the young animals in the BPA50 group. So no gene expression fold changes were observed in the older animals. It was just found in the younger animals. So the conclusions just for my molecular component of the project was that the diethylstilbestrol treatment group, so that positive control group, demonstrated consistent downregulation in male adrenal gland glucocorticoid receptor gene expression at both time points. So both in the younger animals, postnatal day 5, and the older animals at postnatal day 15. So this kind of showed that BPA sensitivity was developmentally stage specific. And our unanticipated result was that 18S rRNA, so this was supposed to be one of our reference genes, was actually sensitive to BPA in our younger animals. So this was a really unanticipated finding from the study. So one of the last things I wanted to mention was the importance of dose response curves. So endocrine disruptors are regularly linked to non-monotonic dose response curves. So it's not a linear curve. But policy has been formed using monotonic curves where the dose makes the poison. So the less amount of exposure isn't as harmful for you. But we're finding out with a lot of endocrine disruptors like BPA, these act in a non-monotonic fashion, meaning that lower levels of exposure can be just as impactful as higher levels of exposure. So this is something that's really being looked at closely in endocrine disruptor research right now. And just a few fun facts before we end. So there's some controversies in the field of BPA research. So this low dose effect, these low dose exposures, non-monotonicity, and source of funding. So source of funding may influence study outcomes. So for the Chapel Hill BPA meetings, they found that over 100 government-funded studies didn't find any adverse or found adverse effects due to low dose exposure to endocrine disruptors, whereas 11 industry-funded studies said that they didn't find any adverse effects. And it was these industry-funded studies that ended up informing policy. So because of what happened with BPA in this Chapel Hill meeting, this is actually what promoted conflict of interest statements to be mandatory on scientific research articles. So that's just an interesting fact. And be mindful of greenwashing. So consumers are often unaware that BPA free doesn't mean estrogen free. So most products that are labeled BPA free still actually have estrogenic activity, and they'll just replace BPA, the A in BPA, with something like S or F. And plastics labeled BPA free have been shown in a study actually to have had the highest amounts of estrogen leached out of them. This is a study by Yang and his colleagues. This is a really interesting study. So it just goes to show that to really ensure that you're avoiding these chemicals in plastics, your best bet is to just swap out plastics completely if that's possible for you, and instead choose things like glass containers, glass Tupperware, and avoid using plastic for your water bottles. You can use even stainless steel. That's a good one too, but just to know with plastic, you'll never be totally sure if you're avoiding endocrine disrupting chemicals that have estrogenic activity. So that's your best bet. So I hope you found this presentation interesting and informative, and please let us know if you have any questions.